This protocol is significant for two main reasons. Number one, it allows a user to be able to determine the cell cycle states of skin cells that have been isolated from an animal model. It also allows us to be able to analyze protein expression in the discrete phases of the cell cycle. Compared to previous methods of determining cell proliferation, our method permits a more precise determination of the different phases in the cell cycle, and we can also analyze greater than 40 different markers in the various stages of cell division. This greatly enhances the application and versatility of the method.
This protocol may be used in cancer research and in any other type of disease where there is a need to determine cell cycle-specific protein expression patterns. In addition, this protocol can be modified to other cell types and in other model organisms. The first-time user should focus on getting the best quality cell preparation possible. This requires the timely processing of experimental skin using fresh reagents, minimal tissue digestion time, and careful disposal of the supernatant after centrifugation to preserve the integrity and quantity of cells.
To begin, design a metal-tagged antibody panel using a free online panel design software as described in the manuscript. Then prepare an IDU stock solution by dissolving IDU powder at 10 milligrams per milliliter in 0.1 normal sodium hydroxide at 60 degrees Celsius. After aliquoting IDU stock solution into microcentrifuge tubes, freeze them at minus 20 degrees Celsius for long-term storage. Immediately before use, adjust the pH of the IDU solution to 7.5 with 12 normal hydrochloric.